lesson is part of a fun podcast-style series created by the Rubicon Bridge team for IGCSE learners everywhere. Keep listening and learning.
Welcome to the Deep Dive. You know, it's actually pretty wild when you stop and think about it.
Mm-hmm.
Every single living thing on this planet, I mean, from the tiniest bacterium you can imagine, right up to the biggest whales swimming in the ocean, they all share this fundamental building block. And today, we're diving deep into that incredible, tiny, absolutely essential powerhouse, the cell. So our mission for this deep dive is really to explore the amazing structures, the huge range of functions, and honestly, the fascinating history of these little microscopic organizers. We've got a whole stack of sources here, articles, some cutting-edge research, plus our own notes, and we're going to unpack the most important bits, the real nuggets of knowledge, and maybe some surprising insights for you. Think of it as your shortcut to really getting why these tiny units are just so fundamental to everything.
And that really hits the nail on the head, doesn't it? I mean, despite being so incredibly small, cells are the true orchestrators of all biological complexity. They're not just tiny parts. They hold the actual secrets to life's endurance, you know? That resilience that's allowed life to thrive here on Earth for billions of years and adapt to almost every environment you can think of. It's just an astonishing level of organization packed in there.
Okay, so let's unpack that a bit. When we talk about cells, like, the core definition is they're the smallest units that all organisms are made from. Simple enough, maybe. But we need to distinguish, right?
Mm-hmm.
Between unicellular organisms, just one cell doing everything, like bacteria, and then multicellular organisms, like us, or plants made of millions, even trillions of cells all working together.
Exactly. And that distinction is key. But underlying both is that really remarkable principle. All new cells come from cells that already exist. It's not spontaneous generation. It's this continuous lineage, this incredible process of replication. It underpins all growth, all repair, all reproduction on the planet. It's more than a definition. It's like a fundamental law of biology.
That continuity idea.
Yeah.
Yeah, it's kind of mind-boggling. Okay, so imagine we could shrink down, like way down, and take a tour inside a typical animal cell. Was the first thing we'd bump into. What are the key structures? Like stepping through the outer edge. That'd be the cell membrane, right? This thin, sort of flexible layer around everything inside. People call it a bouncer, which is a fun analogy, but it's maybe more complex than that. It's partially permeable. That means it controls what gets in and out. Why is that so critical?
Oh, it's absolutely vital. That selective permeability is essential for keeping the cell's internal environment stable, what scientists call homeostasis. Without it, the cell couldn't control, nutrients coming in, waste going out, or even how it communicates with its neighbors. It's not just a passive wall. It's active, constantly sensing, constantly responding.
Huh, okay. Active sensing.
Yeah.
I like that. So once we're past this selective gatekeeper, we're in the cytoplasm, this clear jelly-like stuff filling the cell. You mentioned it's mostly water, maybe 70%. And it's where a lot happens. What kind of happening are we talking about?
Yeah, think of the cytoplasm less like jelly filler and more like a bustling factory floor. It's where countless chemical reactions take place, what we call metabolic reactions. Mm-hmm. All the processes that keep the cell alive, breaking down food molecules, building new parts, sending signals around. It looks like chaos, but it's incredibly organized chaos, making sure everything happens where and when it needs to.
Okay, a bustling, organized chaos. And navigating through that, we definitely spot the big one. The nucleus, often called the cell's control center. And that's because it holds all the genetic information, the DNA. How exactly does this DNA run the show?
The nucleus really is the command center. Inside, the DNA is packaged into these structures we call chromosomes. And within the DNA are specific segments called genes. These genes are basically the instruction manuals for building proteins. And proteins do almost everything. They build structures, speed up reactions, carry messages, transport things. So by controlling which proteins get made and when and how many, the nucleus essentially directs all the cell's operations. It dictates the cell's identity, its function. The sheer amount of information packed in there is just staggering.
Staggering is right. And speaking of doing the work, let's talk about mitochondria, these sort of oval-shaped things, famously the powerhouses of the cell. What makes them so powerful? And I think there's something really interesting about their history.
Absolutely. The mitochondria are where aerobic respiration happens. That's the process where glucose sugar is broken down using oxygen to release a huge amount of energy for the cell. way more than other methods. It's incredibly efficient. But yeah, the history part is fascinating. Mitochondria have their own DNA, completely separate from the DNA in the nucleus. And this, along with other clues, strongly supports the endosymbiotic theory. The idea is that billions of years ago, mitochondria were actually free-living bacteria. They got engulfed by some early ancestor cell, and instead of being digested, they just stayed, formed a partnership. It's an amazing example of how life evolves through cooperation, through merging.
Wow. Parts of us used to be separate organisms. That's wild. Okay, lastly, inside this animal cell, we see these tiny, tiny dots, almost too small to properly see sometimes. The ribosomes. What's their job?
The ribosomes. They might be tiny, but they're crucial. They are the protein-making factories themselves. This is where protein synthesis occurs. They take the instructions sent out from the DNA copies of the genes, and they actually assemble the proteins, amino acid by amino acid. They're incredibly numerous and incredibly important. And yeah, you mentioned earlier, you might also see some small temporary sacs in animal cells, vacuoles or vesicles, usually for like transport or short-term storage.
Okay, that's a fantastic tour of the animal cell.
Ugh.
But now let's switch gears. Let's step into the world of plants. Because their cells share a lot of these parts, but they have some really key differences, don't they? Differences that let them do things animal cells can't, like, stand up straight against gravity and make their own food from sunlight.
Exactly. They face different challenges, so they've evolved different solutions, different structures. While the core machinery is similar, plants needed, well, for one thing, more structural support than animal cells typically do.
Right. And that comes from the cell wall, this rigid outer layer made of something called cellulose. It's outside the cell membrane. Its job is support and protection, stops the cell from bursting if it takes in too much water. But here's a key point. Unlike the membrane, the cell wall is fully permeable. Why is that important? How does that help?
That full permeability is critical. It means water and dissolved stuff, like minerals from the soil, can pass straight through it easily. It doesn't filter them. That lets the cell membrane, which is just inside the wall, do the selective job of controlling what actually enters the living part of the cell. So you get the structural strength of the wall, but the membrane still manages the traffic. And the setup, the strong wall plus the pressure from inside, especially from that big vacuole we'll get to, is what creates turgor pressure. It's what keeps plants rigid, stops them wilting.
Turgor pressure. Got it. Okay, and maybe the most famous difference. Chloroplasts. These little green oval things you find in the leaves and stems. They contain chlorophyll, that green pigment. And chlorophyll is the key to photosynthesis, right? Absorbing sunlight to make food. This sounds simple, but it's basically the foundation for almost all ecosystems on Earth.
It really is. Photosynthesis is the fundamental energy capture process for life as we know it. Plants take light energy, carbon dioxide, and water and turn it into chemical energy sugars. And crucially, they release oxygen as a byproduct, which, most other life needs. Chlorophyll itself is an amazing molecule, perfectly tuned to grab specific wavelengths of sunlight. And yeah, inside the chloroplast, plants often store some of that captured energy temporarily as starch grains. It's just brilliant bioengineering.
Brilliant is the word. And finally, for plants, there's that large permanent vacuole. Animal cells might have small temporary ones, but plant cells usually have this huge central sac filled with fluid. What's its main purpose, other than just taking up space?
Oh, it's much more than just space filler. It's filled with cell sap, that's water, plus dissolved sugars, salts, pigments, sometimes waste products. Its primary role is maintaining that turgor pressure we talked about. It pushes outwards against the cell wall, keeping the cell firm, like air in a tire. But it also stores water, nutrients, and can even help break down waste. It's central to the plant cell's structure and its internal environment.
Okay, amazing differences. Now let's take a big leap back in time. Billions of years. Let's talk about bacteria. These aren't just other tiny organisms. They're a whole different kind of cell, a really ancient form of life. They are unicellular, yes, but the big deal is they are prokaryotic cells. And the absolute key difference, they don't have a nucleus. Their DNA is just elsewhere. What does their ancient history tell us about life itself?
Well, this is where you really appreciate deep time.
Yeah.
Scientists think bacteria first showed up maybe over 3.5 billion years ago. Earth was very different than conditions like modern hot springs, very extreme. And what's incredible is, despite being structurally, simpler, no nucleus, no mitochondria, none of that internal compartmentalization you see in our cells or plant cells, they've been unbelievably successful. They've survived and thrived on Earth almost unchanged in basic form for vastly longer than complex life like us has even existed. Their sheer endurance tells you a lot about what's truly fundamental for a cell to work.
So, simpler structure, but incredible success. What does that basic structure look like then? They have a cell wall, but it's made of peptidoglycan, not cellulose like plants.
Okay.
They've got a cell membrane, cytoplasm, ribosome, so some similarities. But the genetic material, no nucleus. It's a single circular DNA chromosome, sort of floating in the cytoplasm. And sometimes they have extra little loops of DNA called plasmids. What makes them so good at surviving in really harsh places like those early Earth conditions or even today's extreme environments?
That brings us neatly to the extremophiles. Many of these are bacteria, or their relatives, the archaea. They live in places that would just destroy most other cells, boiling hot springs in Yellowstone, super salty lakes, deep sea volcanic vents, acidic pools, you name it. Their success comes down to remarkable biochemical adaptability. Their enzymes work at high temperatures, their membranes can cope with weird pressures or chemicals, and those plasmids you mentioned, they're often key. They can carry genes for things like antibiotic resistance or the ability to eat unusual food sources, and bacteria can swap these plasmids around really easily. So this adaptability, plus their fast reproduction and simple needs, makes them incredibly resilient. They can exploit niches where nothing else can survive. They really force us to broaden our definition of where life is possible.
It's just, astounding. The sheer tenacity.
Yeah.
Okay, so we've looked at complex animal and plant cells, these ancient, tough bacterial cells. But how do we get from individual cells to us, to a complex organism like a human or a tree? This is where the idea of a hierarchy comes in, right? Cells organizing themselves.
Exactly. It's a scaling up of complexity and function. In multicellular organisms, cells aren't just a random jumble. They specialize and cooperate in very structured ways. It's all about organization.
And that starts with specialized cells, doesn't it? Cells that change, that develop unique shapes or features to do one particular job really well. Can you give us a couple of really clear examples?
Oh, definitely. Think about the cells lining your windpipe, your trachea. They have these tiny hair-like things called cilia on their surface. These ciliated cells constantly beat sweeping mucus and trapped dust upwards away from your lungs. That's their specific job. Or neurons, nerve cells. They are often incredibly long and have branched ends, perfect for transmitting electrical signals quickly over long distances in the body. Red blood cells are another great one. They're tiny, they're indented discs, and mature ones even ditch their nucleus, all to maximize space for hemoglobin, the molecule that carries oxygen. And in plants, those palisade mesophyll cells near the top surface of a leaf. They're absolutely jam-packed with chloroplasts, perfectly positioned to capture as much sunlight as possible for photosynthesis. Each one is a specialist.
Masters of their trade. Okay, so when you get a group of similar specialized cells all working together on the same function, that forms a tissue. Like muscle tissues made of muscle cells contracting together. And then if you group different tissues together,
then you get an organ. An organ is a structure made of several different types of tissue, all collaborating to perform a specific, more complex function. Think about the heart. It's mainly muscle tissue for pumping, sure, but it also has nerve tissue to control the beat, connective tissue to hold it together, epithelial tissue lining it, all working as one unit. A plant leaf is an organ too, with tissues for photosynthesis, transport, protection. The kidney is an organ, the stomach. Each one is a multi-tissue collaboration.
And the final level in this hierarchy within one organism, that's organ systems, right?
Exactly. Organ systems are groups of different organs that all work together to carry out a major life process. Like the digestive system that involves the stomach, the small intestine, the large intestine, the liver, the pancreas. All these organs coordinating to break down food, absorb nutrients, and get rid of waste. Or the circulatory system with the heart and blood vessels. Or the nervous system. It's this layered organization from cell to tissue to organ to system that allows multicellular life to be so complex and capable.
It really is an amazing system, from the microscopic level right up to the whole organism. But hang on, all this incredible detail. How do we even know this stuff? Cells are unbelievably small. How do we see them? Which brings us, of course, to microscopes. For a long time, the main tool was the light microscope. Uses visible light, passes it through glass lenses to magnify things. Let us see basic cell shapes. The nucleus, maybe chloroplasts, a good one might magnify, to 500 times or so. The pictures we get are called photomicrographs. This is the first window, really. But to see the real detail inside, the mitochondria, the ribosomes, all that intricate stuff, we needed something much more powerful. That's the electron microscope. It doesn't use light. It uses beams of electrons. What kind of jump in magnification did that give us? What did it let us see?
Oh, the jump was revolutionary. just immense. Electron microscopes can magnify things hundreds of thousands of times, like up to 500,000 times or even more in some cases. Suddenly, we could see the internal folds inside mitochondria, the tiny dots of ribosomes actually making proteins, the complex membranes within the cell, things that were just blurs or completely invisible before snapped into focus. These images, electron micrographs, they didn't just confirm old ideas, they revealed whole new levels of complexity. It completely changed cell biology.
A reminder of how technology drives discovery. And understanding magnification itself is important when looking at these images, isn't it? It's not just making it bigger.
Absolutely. Magnification is fundamentally a ratio. It's the relationship between how big the image appears, say, in the microscope or on the micrograph, and how big the object actually is in reality. Calculating magnification allows us to put a number on it, to quantify just how much bigger we're seeing things. It lets us measure structures accurately and get a true sense of scale within that microscopic world. It connects the image back to reality.
What an incredible journey today. Seriously, from these tiny, tiny units of life just buzzing with complex machinery, right up through how they organize into tissues, organs, entire beings, and the amazing tools that let us peek into their world. It really is humbling when you think about the complexity, the diversity, the sheer ingenuity packed into every single cell, the foundation of everything alive.
It truly is, and it leaves you with a really profound thought, doesn't it? Thinking about those bacteria again. If life started with simple cells like that, billions of years ago, and those simple, incredibly resilient forms are still thriving today, often in the harshest places on Earth, well, what does that tell us about the absolute fundamental requirements for life? What are the core enduring principles? And maybe, just maybe, what lessons do these tiny survivors hold as we